everyone. Before we get started, I have a couple housekeeping notes. So please silence your phones. Photography of ABRF meeting sessions is permitted, subject to speaker's request not to capture pre-publication information, and recordings of the program sessions will be available on the meeting website following the annual meeting. ABRF's social media policy is that everything can be shared. If a speaker asks it not to be shared, please respect the speaker and do not post on any social media. And the hashtag for this event is ABRF 2026. Unless noted by the speaker, questions will be held until the end of the session. Attendees may ask questions at the microphone in the room. And download the ABRF meeting app to view the full schedule, access lightning talk and poster abstracts, connect with other attendees, sponsors, play the ABRF game, and create your personalized meeting schedule. You'll find extra content, including hours of exhibit and text showcases, and thank you to our sponsors. All attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. Hello, and welcome to the Course Communication Building Better Science Communication in Course session. My name is Laura, and I created this session. Today I am joined by Dr. Taylor Edwards and Dr. Yanis Valchos. Hopefully I said that right. Okay. Dr. Taylor Edwards, is the manager of the Arizona Molecular Clinical Corps and the Corps' operations manager at the University of Arizona. He has worked in the University of Arizona core facilities for over 20 years and has extensive experience in managing facility operations and overseeing large interdisciplinary projects within cores. He has worked with core facility personnel ranging from high school students all the way up to doctorate level scientists and bioinformatics. He also seeks to promote science and encourage the public through outreach with community and organization presentations, as well as National Geographic expert for National Geographic expeditions. And then Dr. Yanis Valchos is the director of the Spatial Technologies and Bioinformatics units within the Precision RNA Medicine Corps at the Beth Israel Deaconess Medical Center. He is an assistant professor of pathology at Harvard Medical School and co-director of the Bioinformatics program at the Cancer Research Institute. He is also an associated faculty member of the Harvard Stem Cell Institute. And he has done trailblazing computational non-coding genome research focusing on non-coding RNAs, like microRNAs and long non-coding RNAs. All right. So my presentation is going to be more of a story because I don't have a doctor in front of my name. So let's get started. Okay. So this is me as a child doing science. My path to course is a little different than most people. Science has always been a part of my life. My mom was a pathologist, and I would often go with her to work when I was young. She had like one of those 2-person microscopes, and I could move the little tray up and down, and I don't know. I thought that was awesome. And then we would also go to the lab, and then I could sit there and watch people run around, and I don't know. I felt like part of it. So elementary school was also, I was pretty good at science. I had, the final elementary school experiences, like we got to raise caterpillars into moths, or we got to go to the Pima Air and Space Museum and play around. And the main point for me was that science was easy. I did not have to spend extra time on it outside of school. Unfortunately, that changed for me in high school. After freshman year, I started taking AP classes, and I was not prepared for them. I had to start spending actual energy and time on school, and I was not into that. The worst one, though, for me was my AP science classes. I would work at them, and I couldn't get it. I couldn't grasp the concepts. Even when I put the effort in, it didn't seem to help, and I couldn't understand what we were talking about. My brain kind of just stopped absorbing information, and I was not sure how to handle that. So I kind of just kept everything inside. I was like, something's wrong with me. I can't do this. Science is not for me. Um, and yeah, even though I only took 2 years of AP Science, after high school, I decided that I was never going to do science again if I could help it. So I selected anthropology for my major, which did not have to take any higher level science classes for. And yeah, it was kind of a spur of the moment decision. I wanted to travel and I liked archaeology, so I picked it. Um, this picture is actually of me at a dig. We had an archaeology class at the University of Arizona, and so this is an archaeological site in northeast Tucson that we would go to 3 days a week and just dig. It was pretty cool. So it ended up taking me about 5.5 years to graduate because I did have a double major. And I took summer classes, and so by then I was just over school. So I decided I'm not going to do anything right now. I'm just going to chill. This is my time. Unfortunately, some things happened in the interim. Like my father had a lot of illnesses, he had some health scares, and then I kind of had my own mental illness struggles. And so there were a couple years that passed where I didn't really progress past where I was when I graduated from college. So I was like, I need to do something with my life. And for some reason, I was convinced that I could not do art or anthropology. So I was like, let me go into healthcare. I was comfortable in hospitals, having grown up with them with my mom. And I was especially comfortable with blood and needles. So I was like, oh, I'll be a phlebotomist. Perfect. It seemed to fit everything I wanted. And while it was science adjacent, I did not have to do any more science. So I was on board with that. The thing that I was not prepared for was that just because I was comfortable with blood and needles, other people were not. And they are not afraid to let you know that they do not like what you're doing. Yeah, and unfortunately for phlebotomists, you have to learn on the job. And so it only took a couple of not bad draws, but a couple tries for me to kind of just get very anxious about doing this job. And so after a while, every day I would dread going. And so eventually I quit that too. So basically I was trying to figure out what to do with my life, and when COVID hit. And since I mentioned before that my mom was a pathologist, she had some insight into stuff, and so she was also the director of a clinical genetics lab that was going to take on the antibody COVID testing for the state of Arizona. And she basically said they needed people with lab experience, and I was like, oh, I can do that. I'll do that. She told me all I had to do was to spin down blood tubes. So I was like, fine, I'll do that. I'll sit there and then figure my life out as I sit there and earn money, basically. And so what I knew of labs was that they were in the basement. They were kind of away. There were no windows. And so when I arrived at this lab, I was shocked. It's a lab on the first floor. There's 2 walls of windows and a bunch of science instruments and stuff that I had no idea what they were. So this is the lab as it looks today. It looked a little different when I started, but basically there were all these instruments, there were robots, there were pre and post PCR spaces, and I was like, oh, this is not the hospital lab that I thought it was going to be. So the first day when I got there, I learned that I would not be spinning down blood tubes. I would instead be transferring serum from already spun down tubes into a stock plate. So this is how we were going to do the COVID antibody testing. And I was again pretty nervous. I was like, oh no, this feels like AP Science all over again. I don't know what I'm doing. So my plan was just to pretend like I knew. I was like, I don't know what people know when they come in here from a science background, so I'm just going to know it all. And if I don't know it, I'll look it up later. Thankfully, I was not the sole person transferring serum, so I could watch other people and kind of figure out how to work the manual pipette, which I had never used before. Honestly, the hardest part for me of this whole process was getting the little plugs out of the blood tubes. I spent the entire first day of working there trying to get them out, and I ended up getting blisters on my fingers because I was like, I'm not going to ask for help. I can do this. So basically, the system was once you got all the plugs out of the blood tube, we would place them back on the rack that they came from. We would put like a much easier to remove cap on. We would scan them into our limb system to make sure they were all in the correct location. And then once it was confirmed that the samples were in the right place, we would then transfer the serum into a corresponding stock plate, column by column. And then the plates were foiled and then put in the oven for an hour to kill any live virus. And honestly, the pipette was probably the most challenging thing I had encountered so far. It was hard to get all of the serum out of the tube without hitting that like little plug in it because heights varied, and so I spent the entire summer basically doing that. Every day I would come in, I would transfer all the racks we had into stock plates, put them in the oven, and then go home. So I was pretty good with that. It was nice. I got paid, and then I got to go home for most of the day. After a while, I was asked to pull positive samples because at that time, most people had not had COVID, so we needed positive samples. This is one of those serums. I think it might have been one of the highest positives we had, so he got a little crown. And then towards the end of the summer, I was told I could stay on, but now I would be working until 5. So in the morning, I would transfer my serum, and then in the afternoon, I got to help develop a new method on the robot. So when I thought of robot, I did not picture this. I pictured like a humanoid robot or really anything but this. To me, this was not a robot. This was just a weird, machine that could move around. So I was pretty intimidated by it, honestly. And I was very nervous to start, but thankfully I just had to sit there and watch. And let's see. When I was a phlebotomist, I got to do some lab work. So I got to get cultures ready. I got to get stuff to send out ready, and then I got to stay in slides. But plates were never a thing I had come across. And all of a sudden, there were so many plates. There are big plates, there were small plates, there were plates with a lot of little holes, there were plates with less holes. I had no idea what I was looking at. Luckily, when you develop a method on the robot, you have to start by figuring out which labware you want. And so as the labware got defined, I got to see what they were and kind of better understand what they were used for. Also at the same time, we were working on translating an existing protocol to work on the robot, and while I mainly watched on this, I did get to ask a lot of questions about lab terminology and things that I was just generally curious about. And there was a lot I did not know. Thankfully, the person who was actually developing the robotic methods was kind enough to answer all my dumb questions. And so basically this is when I truly started to relax a little and learn about the lab. Some big things I didn't know was that the fridge was not a fridge, it was a 4C, and that the freezers were not freezers, they were -20s, and then -80 was a whole different thing. And I also learned some things that I had thought I knew, but did not. So I don't know if you ever have to roll your foil onto your plate, but those little things in art are called brayers, and you would never use them like that. It was a big, big change. And I also thought that annealing was a way to make metal more malleable for silversmithing. So while I didn't get to do a lot of hands-on work during this time, I enjoyed it a lot. It was kind of like having a one-on-one tutor for the lab, so I got to ask questions, listen to Twitch streams, and then just generally learn about the robots. It helped to make me feel more comfortable in the labs and prepared me for the next step. So as the fall progressed, I moved from being like an afternoon shadow to now starting work on the COVID ELISA team. Before I started in the lab, people at the U of A from several different labs have been working on developing a high throughput robotic COVID test. They worked on this tirelessly, and basically by the summer of 2020, they had a working robotic test. So I joined the ELISA team when the method was up and running, thankfully. I did not know what an ELISA was. I had never heard of it. I didn't know how it worked. I just knew this is what I was going to be doing now. So in the mornings, I still transferred the serum, but now I did ELISAs in the afternoon. There was a lot to learn. ELISAs are I don't know. There was a lot to learn, basically, and I was not honestly sure why I was there. I was like, I don't know if this is for me. I just transfer the serum. And so I still kind of had those doubts when I first started, the same doubts I'd had before with my other science classes, that maybe I couldn't do this. This was a real test, and we were giving people real information, and I wasn't sure I could live up to the pressure. One of the first things I learned was that I had to take notes. Till this point, I was like, no, my memory's good enough. I'll get it. No. ELISAs had a ton of new things for me to learn. There were a lot of components, and I just could not have kept that straight in my head. So the ELISA reran was over 2 days. Day 0, as we called it, was the day before the run. It was when we would coat the plates with the capture antibody and then prepare everything for the next day's run. Day 1 was the run day of the ELISA, and I learned a lot of stuff. We had to take out the ELISA reagents to make sure they were warm to room temperature on the bench, and then all the samples that we put on these 384-well plates, we had to dilute beforehand. So we had a separate robot that would do the dilutions, and then we would add those solutions to our ELISA plate on another robot. The tricky part was starting the dilutions early enough so that by the time we got to the add serum or add sample step, that was done and we could just put those plates then on the robot. At that time, the method was maybe around 5 hours, so we didn't want to delay it if we could help it. The nice thing about this method was that it was all figured out. We didn't have to do anything. Well, we had to do some stuff, but we didn't have to worry about pauses or anything like that. They came already built in. The main thing we had to do was just make sure that the robot didn't have any errors, and then take things on and add things to the robot, or take things off and add things to the robot when it prompted us. I also had to learn about reagent preparation. Reagent was an official science word, and I was like, I don't know about this. But it turned out that preparing reagents was a lot like following a recipe. My pipetting skills were also important. Before, I had just been transferring serum, so it wasn't that big a deal if I didn't get the full 400 into the plate, but this mattered. And there was a lot more to pipetting than just sucking up liquid and dispensing it. Different reagents had different viscosities. Sometimes it was more important to wet the tip before you use it, and it was best to avoid creating bubbles. All things I would never have guessed. On top of that, reagents were stored at different temperatures, and the plate coating proteins that we used came lyophilized, and I was totally unclear that just meant rehydrating them. We got data from using a Clariostar microplate reader, and those results were analyzed and samples were flagged that could be used as positive controls, like the one we saw a couple back, as well as ones that had too much background noise that would need to be rerun. And then the samples with a high enough positive would be run on an S2 plate to confirm that they were truly COVID positive antibodies. So after a couple weeks, I was an official member of the team, and I knew how important this test was, so I did not want to mess it up. And so I was very, very tense and very anxious, and probably not the best person to work with at that time, but I needed everything to go perfectly because I couldn't let people know that I didn't fully understand, I hadn't been doing science this whole time, basically. And so it was going well for a while. We had some run days and some non-run days, but obviously as time started to progress, COVID became a much bigger deal, and so we got busier and busier, and we had more ELISA plates to run each day. We also got a CDC grant, and so they started sending us a number of samples, and there were some days where I would spend the entire day transferring samples and still have to stay late to complete it all. And soon, now we were running multiple robots with just me and one other person, and it was a lot to do. We would do the dilutions and the run method, and sometimes that meant we would have to stay late to do all the other work. We couldn't complete while the ELISAs were going on. So basically, we needed help. We ended up adding 3 additional techs to the COVID team, and I had never taught anyone how to do something in the lab, so I wasn't really sure of how to go about it. I still felt like an outsider myself, but we needed the help, so I decided, let me teach them the things that I would want to know when I was starting out. It took some time to figure out how to teach 3 new people a skill. It's surprisingly time consuming, and you have to balance people's interests and their attention, which I was not prepared for. I had 3 new people, and I had to figure out how to work them into the team we basically already had. So with the addition of these team members, we now had a team of 5, which was one of the bigger services, and not only was I a co-leader of the service, but now I was managing other people as I did it. Not all the techs were full-time, so we had to plan around their availability and assign them tasks for each day. This also meant we had to plan what kind of run we were doing that day. So it added, I guess, to the amount of work, but it was a good amount. And then since we had all these other people, I stopped doing as much bench work, and now I spent most of the day on the computer or transferring serum. And so now we had the help, but we were way behind in our workload. The CDC had sent us thousands of samples, and so we started running the ELISA every day on every robot we had. We had 4 robots that could hold 4 3D4-well ELISA plates each, so each day we were running about 6,000 individual reactions. We had deadlines to meet, and so we eventually ended up validating a method where we could coat the plates on Friday so that we wouldn't even miss Monday as a run day for the ELISA. We did this for weeks, which turned into months, and then all of a sudden it was over. The CDC decided to stop funding COVID, and so I went from having something to do the whole time every day to now being the sole member of my team in the lab. Everyone else went their ways or went to another lab, and it was just me left in the clinical lab. This was kind of one of our team building things. It was the wall of shame. Often you would need to remove lids on certain plates because the robot could not take them off themselves, and when you forgot to do that, it would destroy the destroy the lid that you left on there. And so we kind of kept that as like a little team bonding thing. So thankfully, the lab decided to keep me on after the CDC funding went away, and now I was going to take all that knowledge that I learned on running the ELISAs, managing a service, and I was going to create my own ELISA service. And while I was still doing ELISAs, nothing was really the same. I wasn't joining a service now, I was creating one. And all the systems that we'd had in place for the COVID ELISA test didn't really work anymore. So we were basically sort of starting from scratch. This new way of working on ELISAs would be kit-based, since we are a cores lab and we obviously take what the researchers want. And so, it was unlike anything I'd done before. I'd never worked with a kit. Everything was individual reagents that we would then mix in certain proportions, and the kit was a totally new thing. So unlike the COVID ELISAs where we could coat our own plates, a lot of the kits we had were the 96 pre-coated plates, and I learned that you can't, once that's done, it's done. I couldn't do reruns, which I was able to do before. I couldn't try to figure out which solution was best. This was it, and most researchers do not want to buy an additional kit just for you to play around with. So I had to figure out kind of ways of, getting everything ready before I put anything onto the plate. After a little while, I did find some ELISA kits that did not include pre-coated plates, just the components, and so that way I could coat them myself. I could use 384-well plates instead of a 96, which gave me more freedom for reruns and also gave me the ability to kind of save researchers a little bit on reagents. You can do basically the same amount of work that you do in a 96 in a 384-well plate. and yeah, it worked out. And so basically, since I was now using 3D 4-wall plates, I could again use the robot, which was nice because I did not want to pipette into 3D 4-wall plates and do washes and stuff. But we didn't have the whole method before. So this time, since the method had already been figured out, we could just break out the parts that were needed. And so basically we had a system for washing the plates and then one for reagent addition. So now the method was fully customizable. I could pick the plate type that I wanted to use on the robot, the tip type, the volumes, and then in the case of the wash method, how long to wait between like the wash buffer and decanting it, which was really helpful. So since the ELISA service no longer occupied all of my time, I had some free time to help around the lab, and I got to do other things like chimerism and PGX, which were nothing like ELISA's. They required a lot more precision and bench work, and so again, I had to learn a new skill, learn how to operate new instruments, and again, I was anxious about messing things up. But now I had been in the lab for about 4 years, and so I felt more comfortable with learning these skills. I think it just takes time and practice to sort of feel like you can take on new challenges. Taking notes was as important as ever, and definitely necessary because unlike the COVID ELISAs, we were not running these tests every day. Sometimes we'd have months would pass between a run, and so I would constantly have to refer to my notes to remind me. And it took longer to learn some services that way than others. And here's a picture of a logo I got to design for the lab. It's not our official logo because the U of A will not allow that, but you can put it on our lab coats. And yeah, so that's basically me, and thank you for coming.
Thank you, Laura. All right. All right. So I am Laura's manager, running the lab that Laura had her lab experience and is still therein. And I will say from the manager's perspective, Laura is one of those great staff members who was paid attention to detail, asked questions when they needed to know something, and was really engaged in learning about what she was doing so that she could do it better and fully understand it. And one of my prize employees, and just been such a pleasure to work with. But it is important to reflect on that experience of coming into a lab and not necessarily knowing the science or having the experience to do that. For myself, I can relate to it because my undergraduate degree was in zoology. I would say the first part of my career was as a wildlife biologist, and then when I went to grad school, I decided to start using DNA to study tortoises because they just lived too long to follow around in the field for 50 years. And I was doing population genetics, so I sort of jumped from a field biologist to a lab biologist without ever really knowing how to be in a lab. And that started in cores. At the time, I could rent space and a bench and a core, and that's where I learned how to extract DNA, and then really the stuff that I'm still doing today. And when we think about science communication, it can be from that starting point, entering the lab, student workers, it includes our full-time staff that we hire in the lab, the graduate students that we work with, and then the outside clients, the core users, and the public as well. So this is sort of a broad topic for the subject today. I'm going to be focusing on the internal science communication that we can do with our staff so that they're better prepared for their jobs. In our group, we have our board of past employees. I've been with the cores for over 20 years. We've done some very large interdisciplinary projects. COVID was one of those. I quadrupled my staff, and we had team members like Laura come in. But we also did a large National Geographic genealogy project for many years. And so we've gone through a lot of people from different backgrounds and different experiences, some staying in cores and some going to other places, and they all need to sort of learn the science behind what they're doing in the lab to be good at it. An example of that is for the genealogy project, we would receive hundreds and hundreds of buccal swab specimens and tubes. We had to prepare the kits, and then we had to receive the tubes and scan them in and get them sorted. And we used a lot of student employees for that, including high school interns. And we would find mistakes. We had a lot of QC's checks for that, and we'd find tubes that didn't have lysis buffer in them. They were empty. We would find things sorted wrong. We have a lot of steps so that those wouldn't have downstream effects. But as running the project, we realized that it's really important to include your staff in the big picture of what they're doing, not just this little myopic task that seems redundant and meaningless. And so we started providing more science background and big picture information for the staff, and it actually then made those students like, wow, I'm part of something really big. It's really important that all these tubes have the same volume of liquid and that they're in the same order, because we don't want anybody's genealogy to be mixed up with somebody else's. So we started a lot of different strategies for sort of including everybody in the big picture of the projects that we were doing in the core facility. Bringing in those students and staff like myself, people come from a lot of different backgrounds. And to this day, now I have my doctorate, but I mean the last chemistry course I took was as an undergraduate. I've never had molecular or cellular biology. I focused on population genetics, so that's where my skill set is. But we've always tried to have more of a knowledge-based hierarchy in the lab. So we have staff members that know a lot more chemistry or have people that work more in immunology, and even if I'm the lab manager, I look to them for advice and trying to figure out methodology or have questions that way too. So we try to create a lab environment where we all share our knowledge. That way it doesn't always have to come from the top down. And I think Laura's story really points out that when you enter the lab, even if you come in with a molecular biology degree, you're not necessarily going to understand really what's happening in a reaction so that you can do optimization or troubleshooting. I think most biology students now probably graduate, they could tell you what PCR is, but they wouldn't really be able to tell you what you need to change to optimize a reaction or things like that. Another reason for educating staff is just the technology is changing so quick. I'm still trying to wrap my head around single-cell sequencing and how I would apply it to something, even though that's a service on our genetics core at the University of Arizona. And it's one of the things I think that actually kept me in the lab, is it was really exciting to see all these new things come up and to try to better understand them. When I went back to do my doctorate after doing my master's, it was right after next-gen sequencing came out. I knew I wanted to apply that to my tortoise research, and I knew that I just needed to have mentors and learn more in order to be truly able to understand the data, not just how to run the protocols. And then actually the great session this morning on neurodiversity was another way to look at not everybody learns the same way. And when you have a large staff working, you need to find methods that can kind of hit different people's learning styles, and it can't just be sort of one size fits all for that kind of science teaching within the laboratory. So as I said, I'm going to focus on sort of our core staff teaching. Janos is going to focus on the external science communication after me. So we're going to cover that kind of broad section. Ultimately, though, when you have a staff that understands the science of what they're doing and they're knowledgeable about it, they're the ones communicating with your clients, and they're better able to help guide them with the research that they're asking your core facility to do. Laura's been great with that. With Eliza's, she'll meet with surgeons from the hospital who they know so much about their model that they're studying, but they have no idea what an ELISA is, and Laura can meet with them and help explain how she's going to design those experiments the most cost-effective and time-effective way for them. So I'm going to start just with some hands-on activities that we've done over the years in the lab. These are all just examples. I'm coming from a genetics course, so they're going to be in that realm, but they're just examples that you could apply to whatever type of core facility that you're doing. As I said, with our genographic project, student workers weren't able to actually run the PCR tests and things like that for us, particularly those high school students. So to give them that broader knowledge of how everything worked, I came up with some hands-on activities so they could do it themselves, but in a model outside of the actual sort of research samples that we were using. And then I would pair a mentor, a staff mentor, with each student so that that mentor got those experiences Laura did when she started teaching others. It helps you better understand your knowledge of the science when you start to try to teach it and communicate it. So instead of that being just top-down and me doing the whole thing, pairing them with staff on this was a really great plan for it. You can see it's a little bit dated. You can tell when we were doing that project, but I called it America's Next Top Taxonomist. And as with my background in wildlife biology, I have access to a lot of interesting specimen types. And so I would put a specimen in a tube without them knowing what it was, and then I'd give them a small sequence of some part of the genome of that specimen that they would have to then design primers for. The X's in that sequence are the kind of unknown region. So they got to design primers to really understand what primers do in a PCR reaction, see the hairpins and folding and things like that. We had them run PCRs, but actually do a real optimization with varying temperature and magnesium, not just kit-based. So you can see their agarose gels for it. And they got to then better understand like how all of that comes together for these big multiplex reactions that we run in the lab and stuff like that, the work that goes in behind it, and more important, the science of what's actually happening in that reaction so that they can troubleshoot. Having the staff mentors, actually some of them had never done this themselves, so it was a really good, even though they understood a lot about it, this was a great way for them to sort of have a student and then go through this process with them. And we kept it on the cheap. I mean, agarose gels and some Sanger sequencing is not a big process. So they'd extract the DNA, design the primers, optimize the reactions, and then they'd get an envelope after they like sequenced it and blasted it to see what their species was. And again, had some fun different species that we had. And they'd get tubes like with a piece of giant squid in it that like dissolved and is gross looking, and they'd have to extract the DNA of it. So that was a really fun project. We have one of our clinical technologists now who was one of those high school students for 2 years in the lab, then went to the U of A, and we hired him immediately as an undergraduate for 4 years. And he's been working in our lab now for, I want to say, 8 years. But coming out of being an undergraduate with 6 years of lab experience, but also the knowledge behind that lab experience to really jump in and start doing things is great. So those are some success stories and what we try to prepare our staff for, whether they're in cores or in other things. So staff can benefit by, they may follow these prescribed protocols, but by actually learning more about what's happening and doing it themselves. I feel they're getting a better science background in it. Having these sort of side activities, there's no consequences for errors. Errors are the ways that we learn, and I feel that was an important part of this. And then it prepares our staff or the students for real life problem solving, regardless of what they go into in the future, whether they stay in cores or whether they go on to grad school or things of that nature. Including staff in this sort of science communication within our facilities, we would have these staff mini seminars. Those would be sort of our service leads. We have, as Laura described, we might have somebody doing ELISAs and then other people doing PCR-based tests. And by having them share that information with others through a sort of this mini seminar type series, it can really help everybody understand the big picture of all the different services going on in the lab. And it can also focus on just general science theory, not necessarily that specific assay, but theory in general, population genetics, evolutionary biology, things that our clients are applying these data to out in the real world. So these are just some examples of them. Population genetics, we were doing a dog sort of genotyping project, so dogs and swabs. Matt Kaplan, who's in the lab here, you recognize all your slides up here and stuff. I'm going to show some more of those too. Zen and the Art of PCR, Matt gave this great presentation I'll show some slides for, where he kind of took all the components of a PCR reaction and made them characters in the story, you know. Taq polymerase and potassium chloride, and magnesium chloride, and could tell the story in a way that our staff could relate to as they start to really try to imagine what's happening in that reaction. We had one of our staff members ask questions about like, hey, when we get these buccal swabs, like how much of that's actually human DNA or other stuff? And so we said, well, why don't you do this experiment so we can find out? So he sort of led this charge, 700 samples, found a housekeeping gene to normalize, did some real-time PCR, but then used that to present to our entire group so that they could see how that real-time sort of methodology works and then have fun with it. We sort of had a who has the dirtiest mouth contest in the process, which I think Matt won. But it was a great way to show, again, for somebody who might run a lot of RT-PCRs, what our clients might be doing with those data by normalizing to that housekeeping gene in very simple kind of ways. So, a 5-minute presentation can actually give people a lot of knowledge on the usefulness of some of our technologies. Other things that we would do, and we still use to this day, are these sort of very simple metaphors in explaining things. Think of your genome as a set of books, like the science library. But then when you extract it, it's like ripping all the binders off and throwing all the pages in a big pile. And then we just want to look at one paragraph in there and just look for a single change in a letter. When we started doing these kinds of like approaches toward it, instead of keeping it super technical, we're still explaining the science, but we're explaining it in a way that's very relatable. I will say I've gotten worse the more knowledgeable I've become or educated in this field as opposed to when I first started doing genetics, because now it's very easy for me to say like, well, a trinucleotide short tandem repeat, blah, 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 blah. When I first used to explain it, it'd be like, think of them like little train cars connected to each other, and sometimes there's more, and sometimes there's less. And we still rely on these simple PowerPoint presentations to explain things, and what we notice too is our lab staff, we all tend to start using the same metaphors if we're giving a tour or talking to a client about how things work. And again, things like PCR, showing the primers, starting and ending primers so you can capture that paragraph. And then these are some images of some of Matt's PCR Zen, where he has all the characters in there, what goes in the master mix, what's going on during the extension, what happens with a low annealing temperature. And then I love how his little Taq polymerases here have the bug eyes because you have so much magnesium. It's like they're over-caffeinated and they just want to start building wherever they can land, and it's easier for them. And while there's a lot of internet resources and YouTube videos that can be very technical about these things, sometimes creating a simple PowerPoint like this yourself helps you find the vocabulary to explain it to others who aren't as versed in it as you might be. So we still rely on these. I think when we trained Laura to do PCR for our chimerism assay, I think I just pulled this up on the computer screen and we started talking about what's going on in those reactions. So these sort of staff presentations have benefits. They prepare your staff for conferences and that professional development. It broadens the knowledge of all the core services to the staff that are working maybe in niche areas, but it's great for them to understand more about what the other services are. Using these sort of metaphors and simple explanations, I think, are key to sort of giving everybody the same vocabulary to work with and understand, and then that they can share with others. And you really might be surprised at how many of your staff who might have been even working for years and doing a PCR reaction or things like that may not have actually gone into it on this kind of level if they just start with kits and things of that nature too. And ultimately, I think it makes us all better scientists the more knowledge that we have with it. We've also then utilized some of that effort that goes into those types of little presentations, no pressure kind of seminars that we put on people, so when they have time and they prepare it, they can submit it. But we use those, and we've started making one-pagers that we'll put on our instruments, which are there either when we're training somebody new or giving a tour of the laboratory, and we can point to a picture. The one on your far left has like the silica bead for magnetic robotic extraction. We have the like DNA stick to the bead, and then we can pull it off with a, pull the beads out with a magnet, and little simple diagrams like that to help explain what we're actually doing in the lab. Barbara Franzway runs the genetics core at the University of Arizona. I love on her website that she has things like ways to assess PCR efficiency or that sort of normalization. I actually had a postdoc this week come in to us. We have a new Biorad QX600 digital droplet PCR. We're running an assay for the Cancer Center. This is a postdoc who's very familiar with PCR and was running his own real-time assays on the same targets. And he said, hey, my results are really different. But he gave us the DNAs. He had used an nanodrop to quantify them. We used a qubit. We normalized ours. The digital droplet PCR is very quantitative, but he was running them based on what he thought those concentrations were, and he was getting differences, and he didn't have a housekeeping gene. So even people who have had that much education and schooling don't necessarily understand all those things. So I think having that outward-facing thing for our clients is helpful when we're generating it ourselves to share with our staff. I personally, again, doing things like the National Geographic tours and stuff like that, I like to read a lot of science books that are written for a lay audience because it gives, even if I know the subject very well, it actually helps me regain those tools for explaining complex things to an audience who's not as familiar with it. Not that these are simple books to read necessarily, but I try, I mean, that's my fun reading. Or TV shows like Cosmos. And then there are a lot of tools, again, for having a diversity of learning options, including online things, YouTube videos, podcasts. This Notebook LM is an AI tool Matt showed me. So it's interesting because you can give it like 15 different scientific papers, and it will spit back the methodologies for you. You can ask an AI to like summarize those in a table, but Notebook LM will also read it back to you in a podcast. That's very weird. And I will say I fed it one of my more theoretical papers, and it didn't get it quite right, but it is a tool, and for thinking about the diversity of ways that people learn, we should consider all those kinds of options out there like that too. And again, as I said, providing that science learning and teaching for our staff really gives them the tools to be better science communicators themselves, and that means our clients that we're trying to, with the clients that we're trying to serve at our core facility. So thank you very much, and at the end we will take questions, but now Giannis is up.
Yeah, good morning from me as well. Here you go. Just give me 1 second. Great. First of all, I wanted to thank our chair, Laura, for putting together this session. You know, I think science communication is an important tool. I will discuss a little bit how our core is wired, and every core uses it differently. But you know, through I think that the combination of hearing Laura and her manager, you saw that Laura's progression was thanks to very inspiring mentorship, and now Laura is a mentor. So good science communication within a core can change people's lives, and can not only keep people, but also, you know, help them grow and retain talent. So I will discuss this a little bit. In our setting, just like 10 seconds about us. We were established back in 2021. We started as a whiteboard exercise in 2020. We became Beth Israel Deaconess Medical Center Institutional Core. Last year, we became the single-cell, designated single-cell spatial core across the Harvard Cancer Center. So what this means, we serve Brigham and Women's, Mass General, Beth Israel, and Boston Children's Hospital's cancer centers as internal. So I have more than 1,500 labs as internal. And we were established with support from the state 5 years ago to become an outwards-facing center. Like everybody can go regardless of where you are. So a big volume of clients that we serve come from pharma, startups, biotech in Massachusetts, but also we have now clients across academic or industry across United States. So we usually talk about the stuff above, what the instruments we have, but today I think the interesting thing for what I'm going to say is that we have internalized through the years the whole process of single-cell spatial. We process the tissue, we do the histology, the TMAs, everything. We do the assay, the sequencing, data analysis as well. And that helps our training and science communication a lot because you have visibility if you want to, and if you invest time in it, you have visibility across the life cycle. So science communication in a core, we heard from the previous discussions about a lot of it about the internal. An internal is this scientific exchange between team members, between a mentor and a mentee, between the questions of a mentee to the mentor, top down from the leadership and bottom up. But also, as you pull in these people to the other aspects of the project, there could be about findings and discoveries. I will talk majorly about the client-facing component, and there is a lot of science back and forth with our clients when you start the project, when you do the project, when you discuss findings. And then I will discuss about the external component, which for me is very, very important. Maybe something that you don't know is we're not a subsidized core. We have a $0 budget from the institution, so we run as a business. We cannot make profit because we're an NIH designated core, but which makes it an even more complicated business versus if we were a for-profit. And everything I will say you already know. But I think the purpose, like how we do things purposefully, maybe that might be of interest to you. So what we're trying, and I will discuss about how scientific communication is a good tool for outreach and marketing. Marketing is a different session at ABRF, and I think a very, very interesting one. But I will show you how science communication can be a part of that. What we want to avoid is to become the science-secluded core. People give us samples, we run samples, we generate data, take data back, the science stays with you, and I don't want to know. You like a commercial CRO, you check the buttons of what the assays you want, ship samples, take data back. You don't talk to anyone. We don't want to be that. And there is a slippery slope because if you're very busy, you want to be that because you want to, end your day faster. So, but we don't want to be that. And the inspiration that we don't want to be that started back in 2021. Initially, we didn't want to internalize anything. We wanted to work with histologists, we wanted to work with sequencing, and we wanted to outsource things. Working with pharma and large consortia, we had to internalize this so we can have control over timelines and quality and accreditations and things like that. So initially, we worked with a phenomenal part-time histotech. She was very, very experienced, and we told her, we're new, we're going to do things differently. Name any wish you want. Yeah, and I was thinking, like, probably higher rate per hour, or maybe go to workshops. Her request was, can I see, you taught me these complicated techniques, and I will be spending half my day to put 3 samples on the slide versus 1,000 for the same amount of time. Can I see what I did? Can you show me what this enabled? Can you show me the results or something when it came back? That's so easy, to do, and that rewired our thinking a lot. So we want to go from running blind, we just run the assays to this purposefully done, and every member sees findings and progress. And people who understand science do better science. That if you understand what you're doing and why you're doing it, 100% you're going to do this better. And then you also have ownership. If you see the paper coming out with your name or just an acknowledgement, of the center you're in, depending on the level of contribution, you still see yourself in this. I did this, this figure is me. Some people might say, yes, Yanis, but you guys do also the combio part. So it's easy for your team to be part of the result process. But if you think about every bioinformatics core you know, and every unhappy PI working with that bioinformatics core, one of the main complaints they have is, I talk to bioinformaticians that they don't care about the science and why I'm doing this. They only care about doing the analysis, but they haven't connected with the scientific component, And I wish bioinformaticians paid more attention to the science and not the tools and things like that. So I think every step of the process, from the project design to the experiment to bioinformatics, can gain from this scientific, participating in the science of the project. The client-facing part, we do this all day, and you discuss science with the clients all day. But if you see this as a tool for, increasing your success rate or revenue or outreach, I think is an interesting case. So in our facility, you do not, even if you work with us 5,000 times and you're one of the first people that worked with us and you have 1,000 projects, you cannot escape the obligatory 1-hour consultation when you start a new project. You have to. You have to meet with people, and you will spend 90% of that hour discussing the science. You will show slides or manuscripts or what you want to achieve. We will not discuss any assays. We'll not discuss, and in the last 5-10 minutes, we will ask about constraints, time, budgets, sample types, volumes, things like that. And then we'll take this information and we'll sit down with a team, and internally we will say, what is the best way for them to answer the question that they have. It could be obvious, but we will spend a little time so we know everybody's on board and everybody's opinion has been heard. And then we return to the client and tell them, based on these priority goals and primary questions and secondary questions, the best assays for you are this, 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 and this. But those match your constraints and your turnaround time and your budget and whatever. And we provide different options. So what I think is interesting is that consultation, apart from increasing your success rate, which is good, if you understand what they're trying to do, we don't care if they come and shop something, and if they select the wrong assay, they will be unhappy. Even if the assay was successful, even if the price was great, if it doesn't provide them what they wanted for, they will be unhappy. So we want, high success rate as a project, not as an experiment. And that's what we report every year, how many led to a manuscript, how many led to a grant. That's nobody asks how many successful runs you did, at least for us. They ask how many led to something that affects the institution. But also increase your credibility. If you start discussing with the PI and ask scientific questions, they say these guys care, but also I think it increases the appreciation, even if they don't work with you because the assays don't fit their budget. They will leave and they will potentially understand what these assays can do next time they write the grant, or that these guys care about my science, and I will tell this to other people that, go and talk to them, they're good. So it is a marketing tool. It is a tool also to increase success rate. And also it is a tool, if you involve a lot of people in that first consultation, everybody in your core will know what this project is about. So you don't need to explain it to anyone. They were there during that presentation. But that is an investment. It's not free. If I have 4 or 5 people or 3 or 4 people in that consultation, that's 3 or 4 work hours that they're not recovering their salary. So for me, because I'm not subsidized, I have to, weigh this. But I think it pays off. The more projects people listen to, the more they understand what people are trying to do. And they can also mix and match projects. Oh, you could also do this in your project. They heard about this from a previous conversation. And then they become better at it, and I don't need to be present as well, and that saves my hour as well. And then when you're doing the project, there is a scientific iteration. The results come out. They talk to the bioinformatics team. The bioinformatics team might involve the experimental team, and then we see new things, and then the PIs might have more questions and more things down the road. And there is science discourse. You talk science, they talk science. And usually we do this for manuscript support. And of course you can do the methods writing. Always a core provides the methods. But if you're part of the discussion, you can also be part of where the paper can go. And you might say, oh, in another project, we were able to achieve this, 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 and that. Or I think with the data, maybe I know you want to go this way, PI, but your data really is gorgeous for that. So maybe you do that. Or sometimes we do this for grant support. And if you're involved early on and people know you talk science and your personnel talk science, they could involve you in the design, they could involve you in, and you have more visibility there. So you're becoming a collaborator. And the last part, which was also mentioned previously that I will touch upon, for us, external science communication is a major outreach tool. I think it's the most powerful outreach tool we have, and we try to use it every day. So a problem with a facility like ours, we have new assays every quarter. Even if it's not a new instrument, a spatial transcriptomics assay now has protein. And I want to be able to communicate that. or we have a new version or higher complexity. So, but even if I did a talk every quarter or every 6 months about our core, who would go every quarter? Like who wants to listen to me talk, say the same things plus protein, for every 3 months or 6 months. So there is this attrition. Usually PIs come first time out of curiosity or postdocs, and then after 3-4 years when they think they need it or they got funded to see what's new. But we want it to come every time, because every time you listen to something, you might think of something else. Or so, of course, there are the talks that I mentioned. And every time we're given by the institution or a consortium or anything to talk about our core or our instruments, we will go. Or if a vendor calls us to talk about, we will go, like 100 times out of 100. But we will also be very aggressive on getting scientific presentations. Did we develop a new method? One of our members will go and present it. Did we have a breakthrough? We also have a dedicated budget for tech dev, for technology development, and that gives opportunities for talks. And we also have, we're trying to support our personnel to submit mini grants, like pilot grants, 20K grants, 15K grants, 30K grants, and whenever those move forward, or we have a big project that they can have a small portion of it to do their own science. Even if it's not going to change the world, sometimes it does. Sometimes it's a bigger project or smaller. That gives you ownership and agency. So, and also we try to get opportunities for lay audience, or philanthropy, or we do walk-arounds in the facility or talk to foundations. Even when you present your science to vendors, what their assay enabled your core to do, vendors want to know that. Your assay enabled us to do this patent, for this group, and to do this manuscript for the other group, or support this core. Vendors want to see impact. And it's also good for their board and also good for their people. And then you can do a quote together or things like that. So how do we do this? We get invited for a scientific talk or any kind of talk, and in the end we'll have 3-4 slides for our core. Everything you saw was enabled by our core. And we have additional things, that new assays, and we're very excited about whatever is coming in the next 6 months. It doesn't cost you anything. It costs you 2-3 slides, 5 minutes, 3 minutes, 1 minute. You can always squeeze in, one in a scientific talk. Or posters. Posters are easier for conferences. So just in 2025, our faculty, in the core had more than 15 scientific talks, different conferences, symposia, institutional events. For institutional means our institution. We went to different retreats, to the core day, which I will have a slide right after, workshops, invited talks, local things. So Longwood is like the Boston Medical, Boston Center, let's say medical school area, where like multiple hospitals are. There was a big event supported by the state and so forth, or we go across the street at the Def-Farber, or Cancer Consortia Regional. Bonnie, hi Bonnie. I didn't know you were coming, but I had a hi for you. Bonnie, his swan song was to do fantastic spatial day at Yale. We were there. We had the scientific talk, and in the end of the talk, or beginning of the talk, there were 2-3 slides about the core. You don't know who's listening, and it's a good outreach tool. And the national symposia, spatial conferences, it doesn't have to be about the core, but the core can always fit there. So the BADMC Core Sciences Day was one of my favorite things last year, and was organized by Vida. Vida is new member in the leadership of research operations in BI. So we wanted to do a core celebrations day in Beth Israel. You have celebrations day for admins, you have celebrations day for everybody, for postdocs. You don't have for cores. But we thought nobody will come. Who's going to come to 6 hours of talks? This is my core, and this is what it does. Why don't they just come and visit and talk to the director, and I don't want to listen to 55 cores? So we did it all about science, and that's why it was Science Day. There were scientific talks in the same session by PIs. There were top multi-core users or high core users, and they were selected by the cores as champions of that core, and they would discuss their manuscripts, they would discuss their patents that they got, and they will say, and the animals were done in the animal facility because of this and reason, and imaging was done there, as they were showing the slides, and spatial was done with, Yanis and the team and so forth. And then within the same session, we had scientific talks from core staff, only scientific talk, and then you have 3-4 minutes to present your core. but the core directors were forced to discuss science. And that was, I think, phenomenal. All the titles in that day were all like, wound healing and this, and that, and cancer evasion and this, and that. So people went for the science and stayed for the core. Does it make sense? We had a panel, how to do more science by cutting down costs if you work with cores early, practically. That's the getaway. And then some people want to discuss one-on-one, with core facilities and stuff. We had a tour with 5, 6 PIs or postdocs, and they could visit the core, and we had people to talk to them, and they could discuss their project and see the instrumentation. And so I loved it. I loved it. So science is the common language we speak. Core facilities are science, are research organizations. We talk science, we do science. And internal scientific literacy, as was discussed previously, enables us to do everything better. Better science, better consultations, increased rate. Client science communication gives you reputation, increases your success rate, gives you agency. And then outreach is a great marketing tool. So because I see this as a good investment. I see this also as part of our infrastructure. It might not be material infrastructure, but I think it's key infrastructure in the core. Increases your project success, your client's appreciation, but also your personnel stays. If they understand the science, they're not a cog, they're not a tool, they're a scientist, and you give them opportunities to grow and stay, and you retain good talent. So what tools I have available? Internal tools. I discuss the project with every member. You support workshops and training opportunities, or you support studies and specializations, or as I mentioned, you can have a small budget for tech dev, where people, if they do this cool thing, they can go and present it afterwards, or if they can have their own project, or you can support grants to submit. There are grants usually open for staff scientists as well. The client-facing component, we involve as many members as are relevant to the project in the consultations. You ask bioinformaticians from your team to, when they respond, they communicate results or questions, to CC the experimentalists that were in the project, so they can still see the discussion that happens on their project. And then it's good to sit down and discuss the science and assays to all members, even if it's evident to you and you think everybody knows. It doesn't hurt, 5 minutes to just discuss, again, the big picture, why we're doing this, what's the goal for this, what's the whole point. And then the external, provide opportunities for talks. Of course, you have to support them going and coming, but there could be a lot of local events that are available. Support poster submissions, that's the best and least painful way for someone who's new to this to start. You have a good poster, people stop by and you talk about it, and I think it's addictive. And also, you don't have to go give a talk for the first conferences. It's also good to support conferences, and if there is a good conference, you might lose a person for half the day, but you could support. I think it's very relevant to the project you're doing. Why don't you go to this cancer symposium that's downstairs for 2-3 hours? You will see why all this is happening. I wanted to thank the amazing people that work in our facility, and for the amazing talent, and also tolerating me through the years. And of course, there will be the shameless slide. If you want to work with us, or if you want to collaborate, we're very collaborative. We work with everyone. If you want to work in this field and you're looking for your next thing, dry or wet, this is our website, QR code, and see how happy everybody looks. Yeah, more than happy to chat with you. Yeah. I wanted to applaud ABR for doing this. I think science communication, we do this all day, and it's an amazing tool, but nobody just, made us stop and think how we do it and whether we can do this better.
Okay, we only have 5 minutes unless you're willing to stay a little bit through lunch, but if you want to ask a question, please come up to the microphone.
Probably I will start.
Because I called you out, right?
Yeah, so I have to ask. So I was just curious, what I haven't heard today was about using some of the new modern methods like Slack channels or dashboards or chatbots, or even using social medias like LinkedIn or similar stuff. So where do you see, do you still see that there is a relevance for these kind of things in academic course, or it's out of our?
Yeah, I wish I had a slide for that. That's a good point. So we do this a lot. So every new manuscript we have with other members or acknowledgement, we have like a LinkedIn post that explains what the project was all about and then how we contributed. It really helps. We don't use Slack, we use River, which is similar to Slack. It's like Slack with project management capabilities. So we have plots and we discuss the project in the Slack, so people, if they want to like remember what this project is about, it's there. We have a lot of project description information in Notion. We use Notion to manage our lab notebook. We have a lot of information there. And what I did last week, and I haven't rolled it out, I created a markup using Claude about the training I want to achieve. I spent half a day. I wrote 5 pages of what the optimal training on a specific topic would cover. And then I asked Claude to make this as a markup document. With the idea is our members have access to Claude, to give that markup to Claude and create a personalized training course for them that meets my criteria, that has these goals set by me, but also follows everybody's pace and questions that they have. But they all have the same course material, but Claude can adjust to everybody's pace, how they do it. Yes, of course, there is not room, but there is, now with the tools we have, it's crazy what you can achieve. I don't know, Taylor?
Yeah, and focusing internally, what I was talking about, I always tell my staff, they use Slack, and I'm not a Slacker, but it is part of the diversity of ways to keep that information out there, and so we certainly try to find as many options as possible. So I appreciate you bringing that up. It's certainly a whole nother avenue to do this with.
Yeah, Slack. My main grievance with Slack is why don't you use Slack? It's very expensive. It's very expensive. If you have a big team, it really adds up. Promo for River, everybody hates it, my group, but I like it. It's very good value for money. And it's River with a Y, if anybody's interested. And I get 5%. No, I'm not.
Thank you. I was just wondering, I love the idea of having that initial consultation with the whole staff present. I think that sometimes can get into a gray area where, like, at what point am I a service and at what point am I a co-author? In your opinion, has that ever gotten contentious? Like, how do you navigate that? Like, those sorts of things?
Yeah, thankfully, Harvard Medical School has specific guidelines. So if you're just doing, I will make it very simple. If you're just doing what you have a line item. We have, for instance, Xenium this, G4X that, single cell this. If people come and shop this after the consultation and it's a successful project and we just do our job, it's an acknowledgement. Of course, they can sometimes elevate you to, like they would say, yeah, I felt you did more than you think. We do not request it. But if we do a new methods development, or if anything that's on the market doesn't work for your project, and we have to sit down and think very hard and be very creative about this, the guidelines show if you have scientific contribution to this, then this is co-authorship, at least based on Harvard Medical School guidelines, and it can be different. We have seen that PIs tend to invite us more to be co-authors in papers. I don't think we have ever reached out to a PI where we felt we had contribution and they didn't. I think they see this collaboration more easily than we think.
It's good to hear. Thank you.
Yeah.
Okay, it is 12, so thank you everyone for coming.